In 2012, West Texas A&M started a research project in which they successfully cloned a bull that they named Alpha from a steer carcass, which was a prime yield grade 1. This is in the very top elite of the USDA's grading system and happens in less than 0.03% of the carcasses nationwide. They also successfully cloned 3 females in which they called Gamma from the same heifer carcass that produced a prime yield grade 1.
We're graders with the United States Department of Agriculture. Today we'll be looking at some beef carcasses and giving you a little bit more information about how the USDA beef grading system is structured. So there's 2 main components to how beef carcasses are merchandised in the United States. It's based on a dual grid system. Half of that grid is based off of a yield grade, the other half is based off of a quality grade. Today, Lauren's going to talk about the yield grade.
So we measure the yield grade with this little ruler. You measure 3/4 of the way up on the ribeye and get the measurement here. And that goes into factors from 1 to 5 yield on a yield grade system. And that calculates the amount of fat or cover that's on the carcass.
The yield grade system incorporates the animal's muscle, their subcutaneous fat, their internal fat, and then all of that is balanced out with how heavy the animal is or their weight. So then on the other side of the grid we have what's called the quality grade system. My job as a USDA grader is to first quality grade the carcass. As you can see here, this carcass is a top choice carcass, so that's obviously a lot more valuable than a select or low choice carcass. We also, as a USDA grader, want to determine the amount of red meat yield within the carcass, so how much muscle is in the carcass, so we can do that by laying a grid on top of the ribeye and then counting the number of square inches that it encompasses.
Hi, I'm Kyle. I'm the owner of Innovative Meat Packing.
And I'm Joe. I'm just looking to buy some meat for my family. Oh, yeah.
So do we find a carcass that interested you?
I think so. I want that clone beef.
Starting the day, tomorrow's a new, and I'm still local enough to choke you to death with a Charleston chew.
While not clones themselves, the 13 offspring of Alpha and Gamma were the first of their kind, as they originated from parents that were derived from cloned carcasses. From there, 7 of these steers were harvested, and 1 of the steers graded prime, while the other 6 remained in the upper 2/3 of choice. Industry-wide, low choice is considered average. So, the calves performed better than average, and within the industry standards, only 5% of carcasses grade prime. Furthermore, all 7 of the steers graded above industry average in terms of yield grade, as they were all 1s and 2s. Industry average is a yield grade 3. Furthermore, the steers all had 16% less fat trim, 9% more ribeye, and 45% more marbling.
With standard reproduction, haploid sperm and egg cells combine to form a diploid embryo, with each gamete contributing half of the new organism's genetic material. Consequently, many different mechanisms in this process create genetic diversity. In contrast, cloning utilizes only 1 copy of genetic material, and the resulting organism shares the same genetic code as its so-called parent.
Somatic cell nuclear transfer is the process by which cloning takes place. A differentiated somatic cell intact with an organism's genome is collected. In this case, a skeletal muscle tissue sample was taken from a prime yield grade 1 carcass. This cell is then placed inside an enucleated oocyte from another organism. Essentially, a diploid cell is transferred to a cell with no genetic material. Once inside, cytoplasmic mechanisms cause genes from the previously differentiated cell to turn on in a process called reprogramming. Likewise, protein synthesis and gene expression are able to occur, and the cloned organism is formed via mitosis. Many unknown complications may lead to embryos not being viable in the process, making it very costly and inefficient. However, this technology has given us the ability to maximize genetic potential of superior animals.
We here at Kansas State love our cows. Thanks for watching.